In January 1976, a 28-year-old venture capitalist drove up from Silicon Valley to the University of California at San Francisco. He had called ahead and asked for 10 minutes with a biochemist named Herbert Boyer. Boyer agreed to give him 10 minutes. The conversation moved to a nearby bar. It lasted 3 hours. By the end, two men had agreed to go into business together, with $500 each and an idea that would change medicine forever. That company was Genentech, and what Boyer and Swanson built together was the entire biotechnology industry. Herbert Wayne Boyer was born on July 10, 1936, in Derry, Pennsylvania. He studied biology and chemistry at St. Vincent College, earned his Ph.D. from the University of Pittsburgh, and joined the faculty at UCSF in 1966. In the early 1970s, working with Stanley Cohen at Stanford, Boyer co-invented recombinant DNA technology, the ability to cut a gene from one organism using molecular scissors called restriction enzymes, splice it into a bacterium, and have the bacterium produce the protein that gene coded for. It was, as the National Inventors Hall of Fame later called it, the most significant achievement in molecular biology since Watson and Crick's discovery of the double helix in 1953. Boyer, wearing his denim vest and bell-bottom jeans, was already thinking about his next experiment. He had no commercial ambitions, yet. Robert Swanson was born in 1947 and graduated from MIT in 1970 with a degree in chemistry and an MBA from the Sloan School of Management. He joined Kleiner Perkins, the leading Silicon Valley venture capital firm. By 1975, Swanson was reading every paper he could find on recombinant DNA and becoming increasingly convinced that the technology could produce medically useful proteins, insulin, growth hormone, interferon, in commercial quantities. He called every expert he could find. Their answer was unanimous. Too early. Biology couldn't be commercialized yet. The science wasn't ready. Swanson didn't believe them. He called Boyer and asked for 10 minutes. Boyer gave him more than 10 minutes. At Herb's place, a bar near UCSF, Swanson laid out his conviction. Recombinant DNA was ready to be a product, a company, an industry. Boyer listened, leaning back in his chair, skeptical but intrigued. He was the first expert Swanson had spoken to who didn't immediately say no. By the end of the evening, they had agreed to try. Each invested $500. Each received 25,000 shares of common stock. Swanson then pitched Kleiner Perkins, who provided $100,000 in seed funding. On April 7, 1976, Genentech was formally incorporated. Swanson became CEO at $2,500 a month. Boyer served as vice president while remaining at UCSF. Today, in the courtyard of Genentech's campus in South San Francisco, there is a life-size bronze sculpture of that meeting. Two men at a bar table, each with a beer. Swanson in a suit, leaning forward. Boyer in his denim vest, leaning back. The moment an industry was born. Genentech had no laboratory of its own in 1976. Swanson subcontracted the science to Boyer's lab at UCSF and two other San Francisco labs, paying them per experiment. The first goal was not insulin. It was simpler. They would produce somatostatin, a human brain hormone, in a bacterium. If they could do that, they could prove the concept. In August 1977, just 16 months after founding, they succeeded. Somatostatin was produced entirely inside a bacterium, the first human protein ever manufactured in a microorganism. The concept was proved. Now they went after insulin. Swanson had chosen insulin strategically. It was a well-understood protein. The market was large, over $100 million in annual sales, and there was a clear patient advantage. All existing insulin at the time came from pig and cow pancreases. Many diabetic patients developed allergic reactions. Human insulin, identical to what the body naturally produces, would be superior in every way. In August 1978, Genentech scientists synthesized human insulin using recombinant DNA. That same month, Swanson negotiated a partnership with Eli Lilly, the world's leading insulin manufacturer, who would fund further development and handle FDA approval. Genentech kept the patents. Eli Lilly provided the scale. This arrangement, small biotech company discovers, large pharmaceutical company distributes, became the template for every major biotech deal that followed. In 1982, Humulin, recombinant human insulin, became the first biotech product ever approved by the FDA. Human insulin, at scale, free of allergy risk, for diabetics worldwide. While Genentech was building toward its first products, Swanson had another move to make. On October 14, 1980, Genentech offered shares to the public on the New York Stock Exchange under the ticker symbol GENE. The stock was priced at $35 per share, above the expected range. Less than a minute after trading began, it hit $80. Within 20 minutes, it reached $89. It was the fastest first-day gain in Wall Street history for a newly listed company. Genentech closed the day at $71.25, having raised $35 million at a market valuation of $400 million. The company had no approved drugs. It had lost money for most of its existence. Goldman Sachs co-chairman John Whitehead said he could not recall a new issue having such a meteoric rise. What investors were buying was not earnings. They were buying a platform, the proof that living cells could be programmed to manufacture medicine. And they were right. Boyer and Swanson each held 925,000 shares that morning. Their $500 investment 4 years earlier was now worth tens of millions of dollars. Harold Evans placed Boyer and Swanson in They Made America because they crossed a frontier that no one had crossed before. Every previous industrial revolution used physical or chemical processes: steel, electricity, oil, electronics. Boyer and Swanson turned living biology into a manufacturing platform. A bacterium became a factory. A gene became a blueprint. For the first time, you could program life to produce medicine, with precision, at scale, reproduced identically every time. Before recombinant insulin, diabetics depended on animal pancreases. Before recombinant growth hormone, children with deficiencies faced a supply constrained by human cadavers. Recombinant DNA removed those bottlenecks, making medicines that had been scarce or allergenic into medicines that were abundant, safe, and human. And the structure they invented, a small science-driven company, venture-funded, patent-protected, partnered with large pharma, became the operating model for thousands of companies and a global industry worth over $500 billion annually. What did they actually build? 4 things that are now permanent. First, the biotech industry itself, with Genentech as the proof of concept that thousands of companies replicated. Second, biological medicines at scale: insulin, growth hormones, cancer therapies, clot-busting drugs that didn't exist or were dangerously scarce before recombinant DNA. Third, the venture capital science partnership, proving that VC could fund biology, which opened universities to commercial collaboration and made the U.S. the world's dominant biomedical power. Fourth, the platform for everything that came next: mRNA vaccines, gene therapy, CRISPR, cancer immunotherapy. When Moderna built a COVID vaccine in days using mRNA, it was building on foundations laid in a San Francisco bar in 1976. 3 lessons from their story. First, the partnership is the invention. Boyer had the science, but no commercial vision. Swanson had the vision, but needed a scientist who believed. Neither could have built Genentech alone. The right partnership doesn't just combine 2 people's skills, it multiplies them. Second, consensus isn't always right. Every expert said it was too early. They were wrong. When every voice in a field agrees something cannot yet be done, it is worth asking whether they are reasoning from evidence, or simply from an inability to imagine the next step. Third, the first product proves the platform. Swanson chose insulin not because it was the most ambitious molecule, but because proving it would validate everything else. The strategic first move matters as much as the ultimate vision. They met for 10 minutes and stayed for 3 hours. That conversation, in a bar near UCSF in January 1976, produced the biotech industry, the VC science model, and the biological medicines that now treat hundreds of millions of people. Thank you for joining us for episode 18. Our next episode is Bill Gates and Steve Jobs, the two men who put a computer on every desk and a computer in every pocket. Until then, stay curious.